friends, today we are going to study about another fibrous protein, that is collagen. In the last video we studied about keratin. Today we'll study about collagen. It provides strength. This is the most abundant vertebrate protein. Collagen is strong. It is insoluble because it is a type of fibrous protein, and as we studied already that fibrous proteins are insoluble in water, and hence the collagen is. Collagens are found in connective tissues such as tendons, cartilage, organic matrix of bone, and the cornea of the eye. Collagen consists of three polypeptide chains. In case of alpha keratin, we studied that it is made up of two polypeptide chains. Here, the collagen consists of three polypeptide chains. The collagen helix, that is the secondary structure, is a left-handed orientation. There are total of 28 collagen varieties found in our body. Collagen consists of 3 amino acid residues per turn. If you remember from keratin, it is 3.5 residues per turn. Here in collagen, it is 3 amino acid residues per turn. This is how the structure of collagen looks like. It consists of 3 polypeptide chains. Now, as we already studied in the keratin one, the secondary structure consists of alpha helix. Two such alpha helix will join and form a coiled coil structure. In case of alpha keratin, two such coiled coil join to form protofilament, whereas in collagen, three such coiled coil will join together. This is the difference between keratin and the collagen. Means 35% of glycine, 11% alanine, and 21% of proline and 4-hydroxyprolyl. Apart from 4-hydroxyprolyl, it also contains 3-hydroxyprolyl and 5-hydroxylysyl, but in smaller amounts. Now in collagen, there is a repeat of a tripeptide unit, that is glycine X and Y. This X is mostly proline and this Y is often 4-hydroxyproline. So in collagen, this tripeptide repeating unit is found. Now there is a hydrogen bond formation between the glycine and the proline residue of the neighboring chain. As we studied, it is of 3 polypeptide. The glycine present in one polypeptide is hydrogen bonded with the proline present in the neighboring chain. Now why glycine is found in this tripeptide? What is the importance of glycine? The thing is, as because 3 polypeptides are super twisted, so that means between these polypeptides very small gap is there. In that gap only glycine can fit because glycine is the smallest amino acid. Its R group is H only. So the side chain of glycine allows it to fit in this tight packing. That's why every third amino acid or every third residue of each polypeptide chain is a glycine. And mostly glycine is found in these turns where two polypeptides are twisted among each other. In that twisting point, the glycines are found. Because in that small gap, only glycine is the amino acid that can fit there. Now, proline prevents it to form alpha helix, that is the right-handed. So that prevention is performed by proline. Again, the proline and 4-hydroxyprolyl, they help in twisting of collagen helix. Because of this tight wrapping or tight twisting of these alpha chains, they provide the great tensile strength to the collagen. Therefore, the collagen in our body, it helps in providing a great support to our body. The food product gelatin, it is mostly a collagen product. Now these non-standard residues like 4-hydroxyprolyl, 3-hydroxyprolyl, these non-standard amino acid residues, these are formed after the collagen polypeptides are synthesized. Now to convert proline into hydroxyproline, the enzyme needed is prolyl hydroxylase, and this enzyme requires ascorbic acid, which is also known as vitamin C. Hence, due to the deficiency of vitamin C, what will happen? This enzyme won't be able to function properly. And if this enzyme won't function, then proline will not get converted into hydroxyproline. And if this conversion won't happen, then all the functions of hydroxyproline will stop. And what are the functions of hydroxyproline? It helps in twisting, it helps in maintaining the stability of collagen, so all will get disturbed. And one known disease due to lack of vitamin C is scurvy. Now, what are collagen fibrils? Collagen fibrils are supramolecular assembly of multiple collagen molecules. So, collagen fibrils are supramolecular assemblies of these triple helical collagen molecules. Now, these are stabilized by hydrophobic interaction. Why hydrophobic interaction? As we already studied, for any molecule, the interior part of the molecule is maintained or remain separated from aquatic surrounding is through the hydrophobic interactions. Now, during this packing of collagen molecules into collagen fibrils, there happens some unusual bond formation. And what are these unusual bond formation? That is a bond that involves a bonding between lysine, hydroxylysine, or histidine residues with X and Y position in collagen. We already studied that there is a repeat of this tripeptide in collagen. So in the collagen fibril, what happens? These fibrils are cross-linked between, for example, lysine and with this X or with this Y. An enzyme needed for this cross-linking is lysyl oxidase. Because of this cross-linking, some unusual amino acid residues are formed. Now, what happens during aging? The connective tissues become rigid and brittle because of this crosslinks. All these crosslinks increases as we ages. So more and more crosslinks will make the collagen fibrils more and more rigid and brittle. And this is the reason why meat from older animals is tougher than meat from younger animals. Because as it ages, the cross-linking will increase. More and more cross-linking means more rigidity. So as we ages, the meat becomes more and more rigid in nature. This is how a collagen fibrils look like. These individual strands are showing collagen molecules. So multiple such collagen molecules bonded with each other through unusual covalent bonds will make a collagen fibrils. Now, because in collagen it lacks cysteine residues, therefore there is no disulfide bond formation in collagen. Now, there are few diseases and disorders that are related with collagen. The first disease is osteogenesis imperfecta. This is an abnormal bone formation in babies. Why it happens? When the glycine gets substituted to cysteine or serine. Similarly, there is one more syndrome, that is Ehlers-Danlos syndrome. That means loose joints. This occurs when glycine gets converted to serine and cysteine again. So from these two diseases, what we can see is the glycine is a very important amino acid in case of collagen. The mutations in the glycine cannot be tolerated. So glycine should be a conserved amino acid in case of collagen. Any mutation in the glycine will affect the structure and stability of the collagen molecule. Why? Because every third residue in a polypeptide is a glycine. And why so? Because in collagen, it is a super twisting of 3 polypeptides. Because of this super twisting, very few space or gap is present between these polypeptides. Such a small gap can only be filled with the glycine residue. If in place of glycine any other residue come in that position, it will affect the structure and the stability of collagen molecule. So therefore, the mutation in the glycine cannot be tolerated and hence it will lead to any disease or disorders. The next example of a fibrous protein is a silk fibron. So silk fibron is a protein of silk. This silk or this particular protein is produced by insects and spiders. It is in beta conformation. These fibrons are rich in alanine and glycine residues. They have an extensive of hydrogen bonding and van der Waals interactions. The silk does not stretch. What does it mean? Because already it is in a beta conformation. Beta conformation is a stretched conformation. Whenever anything is stretched, it converts from alpha to beta conformation. So already it is in a stretched form. So it can't be stretched more. The structure is flexible because it is stabilized by weak interaction. So this is the third example of fibrous protein. So here we end the structure and function of fibrous protein. In the next video, we'll study about globular proteins. Now, one question from my end for you all, that is, explain why gelatin, which is mostly collagen, is nutritionally inferior compared to other types of protein. Gelatin, though it is used in the preparation of many food items, but if we see its nutritional aspect, it is very inferior in nutrition compared to other proteins. So you have to explain why. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.